Hello. What we're going to do in this lecture is we're going to begin our discussion of cellular respiration. Now realize that we want to look at it first as an overview. We want to get a general sense for what the whole story is. Then we're going to take each individual component and break it down and look at the subcomponents. Eventually we'll bring it all back together and talk about it in terms of a summary event again. But initially let's discuss it in the overview. When we're talking about cellular respiration, we're going to be talking about a process where our cells are breaking down molecules. In particular, we're going to focus on the breakdown of carbohydrates, sugars, to produce ATPs. When we talk about an organism like ourselves, we're going to be talking about a process that's called aerobic respiration, but eventually I'll also discuss with you the distinctions between aerobic and anaerobic respiration. Aerobic respiration is going to be this harvesting of energy from sugar molecules. And what we're going to find is that the overall products we're going to make, the raw materials we take in, are the sugar and oxygen. But the products we're going to produce are going to be CO2 that you breathe out. Water is going to be a product of this process, and we should make a relatively large amount of ATPs. That's the goal. We're going to eventually keep track. We're going to have a tally of the ATPs produced from the process of aerobic respiration. Now, if you think about somebody that's exercising, somebody that's carrying out some kind of activity, you know that that person is taking in gases, breathing, and exhaling. Sometimes people refer to that as respiration. That's not what we're talking about. But breathing and cellular respiration are tied to each other. I mean, the whole point of us taking in the oxygen gas is so that we can make use of it as part of our process of aerobic respiration. Later, what we're going to find is that it's a necessary raw material for cellular respiration in organisms like ourselves. And we're going to see it used as, and this term will make more sense later, a final electron acceptor. You might remember that I introduced the idea of chemiosmosis and the need for an electron acceptor. That's the key use of oxygen in our cells. Also, as we exhale, we're getting rid of some of the waste materials that are released from this respiration process. CO2 in particular, and also as we breathe out, we release some of the excess water that we produce from this process of respiration. Remember, the overall event is carried out to make ATPs. So as you look at the summary at the bottom of the slide, you can see that our raw materials are sugar and oxygen. Our products are ATPs, CO2, and water. That's our generalized overview of this process. We can look at it in another way, another overview of the process, and we can see that what we're saying is that we're taking this glucose molecule, C6H12O6, common starting point, not the only molecule that can be broken down by cells for energy, but it is the most common starting point. We take in oxygen gas from the environment. Notice 6 molecules of oxygen gas. We go through a series of chemical reactions. So when you see this one arrow that's located right here, you want to think of that as being a summary of the almost 30 or more chemical reactions that we're going to take a look at later. So this is very, very much a summary of the process. Notice what we're making. 6 CO2s, one of our waste materials. We produce 6 H2Os, water, another waste material. And we're going to generate ATPs. Later we're going to see that we should be able to generate as many as 38 ATPs if aerobic respiration is done and done well by a cell. All right, so ATP production is going to be 36 to 38 ATPs, a fairly substantial amount. You might think of that as being a pretty efficient process, but in reality, it's only about 40% efficient. Notice that if I took some glucose and I set it on fire and burned it, then you could argue that I release all of the energy stored in the glucose in the form of light and heat energy. You could say that that's a 100% efficient process if we're just measuring the energy released from the molecule. Now, you might think of this more appropriately as being like a log burning in a fireplace, because remember, a log is composed of just long chains of glucose molecules, and that might give you a better image to think about in terms of this burning process. Our cells are basically going to be carrying out the same kind of chemical event. We're going to be breaking down glucose molecules with oxygen required and we're going to turn that into the energy, in that glucose molecule. We want to turn it into energy stored in ATP. How well do we do that? Well, we're one of the best organisms at doing that process. Aerobic respiration is one of the most efficient processes in terms of converting stored energy and carbohydrates to ATP energy, but notice it's only about 40% efficient. At best, living things can only turn 40% of the stored energy in their food molecules into energy stored in ATPs. Where does the rest of it go? Heat. Remember what we said about reactions, that there's always going to be some waste energy. And we think of this heat that we generate as being the wasted energy, the energy that didn't go into ATP production. So our 40% efficiency is based upon us being able to make those 36 to 38 ATPs from that one glucose molecule. By the way, that's much better than an automobile. Automobiles are commonly only 20-25% efficient in terms of turning the gasoline stored energy into motion of the vehicle. Much of that energy, 75% of it, is lost as heat, wasted energy, energy that doesn't go to moving the vehicle forward. So we're fairly efficient, more efficient than an automobile at least. Now, there's a lot of activities we carry out that consume large amount of calories. And you can take a look at this illustration and just see that there's a variety of activities we carry out that consume energy, consume ATPs. And so we think of that, we measure that energy in terms of calories, kilocalories. All right? So ATP are providing the energy for all these different muscular activities. Where does the ATP get its energy? From the breakdown of organic molecules like glucose. So the foods we take in provide the energy to make the ATPs. The ATPs provide the energy for the various muscular activities, the whole range of cellular activities that are endergonic, that require energy. ATP provides that energy. So what we're dealing with here is a type of release and storage of energy, aren't we? So think back to what we've already talked about in terms of electrons or hydrogen atoms being moved back and forth between molecules. Remember we discussed the idea of redox reactions, right? Oxidation and reduction. So notice what's happening to glucose. It gets turned into CO2. It's lost hydrogen atoms. We would say that glucose is oxidized to CO2. Look at O2. The result of this process in terms of what happens to O2 is O2 gains hydrogen atoms. O2 gets turned into H2O. So we would say that oxygen is reduced, right, to produce water during this process. So oxidation and reduction, right, those key energy gaining and releasing, storage and release processes are part of this respiration event. Also notice that we do release some of the energy in the form of energy stored in ATP molecules. And of course, we could add to this illustration that there's also energy released as what? Heat energy, right. Take a look at that and think about it for a moment. See if that word fits in your mind in terms of the way I've just described it. So, who gets involved in this? Let's think back to our electron carrier molecules. They're going to be a key part of the cellular respiration event. What's going to happen is enzymes are going to get involved with these coenzyme molecules like NAD, remove hydrogen atoms from the organic molecules going through the steps of this pathway, take those hydrogen atoms, add them to the NAD, the electron carrier, which then in its reduced state, NADH, is going to deliver those deliver those hydrogen atoms to the molecules of the electron transport chain. All right, so once again, we're tying this into those ideas of oxidation and reduction, of electron carrier molecules transporting these packages of energy from one place in the cell to another. Remember, these are the types of reactions we called redox reactions. And in the electron transport chain of the cell, where the electron carriers drop off their load of electrons, those electrons are going to get passed down the chain of molecules in the electron transport chain. They're going to release some of their energy, but they release it in a sense slowly, Contrast this with the rapid energy release of a log burning. There, the energy stored in the bonds of the glucose molecule are released explosively, all at once. In the cells, we can't have that happen. So we have to tear apart the glucose molecule slowly in steps. Pull off an electron here, take off an electron there. By stripping off the energy slowly, we can release the energy in a more metered, controlled way. What's that energy used to do? The energy is used to create that ion gradient we had mentioned before. And notice that those electrons that pass down the chain eventually end up going to a final electron acceptor. And in the case of our cells, that's oxygen. Remember, we do aerobic respiration. Later, when we look at anaerobic respiration, that's going to be the key place where things are different. In anaerobic respiration, there's a different final electron acceptor. Okay? So, why do we use this electron transport chain? Why do we break down glucose stepwise? So that we can release energy in small packages. If all the energy of one glucose molecule was released simultaneously, it would destroy the cell. It would be too much energy released too quickly for the cell to deal with. As a result, the cell moderates the breakdown, chops up the glucose in a stepwise fashion, pulling off bits of energy at different times. We're going to see that when we look at the detailed steps of this respiration process. Here's that idea of that explosive release of energy. Far too much for what a cell could handle. When we see something burning, we're seeing a very rapid process. We're seeing a very rapid process of that fuel molecule. It could be a piece of paper made out of glucose molecules or a log made out of glucose molecules. And we're seeing that glucose molecules, those bonds of those molecules, breaking explosively. We see the molecule experiencing the process of oxidation very rapidly. Remember, interestingly enough, that oxygen is also required for burning, isn't it? Yeah, so it's going to tie in. We want to think of what the cell is doing is it's a slow burn. It's taking the glucose molecule and allowing it to go through the same basic chemical reaction as burning, but in a controlled fashion. Okay? What we're going to find is that we're going to see two basic mechanisms. We've already talked about those. I've introduced these ideas to you. One of them is that we create a hydrogen ion gradient. We use an electron transport chain and the energy of electrons to create a hydrogen ion gradient, use that gradient to power ATP synthase, production of ATPs. If you remember, this event is called chemiosmosis. And you may also remember that we talked about this as part of both oxidative phosphorylation. It's the key process in oxidative phosphorylation. And we also talked about it as a key process associated with photophosphorylation. So given this discussion that we're having right now, you can see that we're going to expect to find a type of chemiosmosis as part of cellular respiration. As a matter of fact, we're going to say that cellular respiration has as one of its key thirds the process of oxidative phosphorylation, a type of chemiosmosis. So we're going to want to make sure we look for that as we go through the individualized steps of this process of respiration. Now, also we said that there was another way of making ATPs, substrate level phosphorylation. And in this representation, you can see what's occurring. I have a substrate molecule bound to its particular enzyme, but while it's going through its chemical reaction, a phosphate group is removed from the substrate molecule and being added to an ADP. So I could say that I'm producing this ATP molecule from energy provided by the substrate molecule. Notice this substrate is also providing the phosphate group needed to make the ATP. So, two basic mechanisms for making ATP. A chemiosmotic mechanism, we found that both in oxidative and photophosphorylation, and a substrate level process in which ATPs are made with energy from a substrate molecule. We're going to want to look for the specific examples of this as we go through the detailed coverage of this process. Let's summarize. We said that one of the key events we're going to be taking a look is an event called cellular respiration. This cellular respiration process can be thought of overall as being a catabolic event. Remember what catabolism is: breaking something apart. What are we breaking apart? Glucose. We're taking a carbohydrate molecule and we're going to chop it up into pieces. Think about what the products were: CO2 and water, carbon, oxygen, hydrogen, the parts of that original glucose molecule we started with. It's overall an exergonic event, because as I break down that glucose molecule, energy is released. Now, what do I use that energy for? I use the energy for making ATPs. So we're going to expect to see ATP production as a part of this event. In respiration, we're going to find both the chemiosmotic event of oxidative phosphorylation and the process of substrate level phosphorylation. Now, those are going to both be components of this respiration event. Remember, we also mentioned that there is that process called photophosphorylation. It's another type of chemiosmotic event. We're going to see that when we have our discussion of photosynthesis. So, respiration, breaking down organic molecules. Why? To make ATPs. How do I make the ATPs? Substrate level phosphorylation, oxidative phosphorylation. All right, it's an exciting event, an important event, and you're going to want to make sure that you feel comfortable with each of the components as we go through them separately. Thanks very much. Look forward to talking to you next time.